All right, I will slowly get going now. People can still pop in as I introduce. So, welcome again to this edition of the Memory Decoding Journal Club, a journal club that we typically do every 2 weeks, and that is working its way towards an event at the Society for Neuroscience annual meeting where the Aspirational Neuroscience Prize will be handed out to papers that are particularly relevant to the problem of, or the challenge of, memory decoding. Today's presentation, I don't know if I can do it justice because this is a little bit of a different paper. First of all, this is a review paper. So there are quite a lot of different things in the paper, and each one of them is touched upon rather than going in really deep. But, I thought it would be worthwhile to bring in this topic of the dendritic engram, that concept, or dendritic clustering and computation, everything that's associated with that, because at least this is an example of something where there is a claim or a proposal that we look at engrams differently, that we don't think of collections of neurons, but instead we think about dendritic branches as being relevant units. And also, I want to apologize for my voice. It's very raspy today. I hope you don't mind, or maybe you have a smart filter on your end that can take that out. I'm just going to get going, and my voice will be what my voice is. So we're diving into the dendritic engram. This is a 2023 paper by Kastellakis and colleagues. And when we talk about memory storage, most of us are used to thinking in terms of networks of neurons and the strengthening or weakening of connections between them. So the twist here is that the authors are proposing that individual dendritic branches, so those fine extensions coming off the neuron, could themselves be fundamental units where memories are stored. And we're going to go step by step through that idea, the evidence for it, some of the computational implications, and a few open questions where the jury is still out. Or maybe I'm just the only one who still has questions about them. We'll see about that. So here's the core claim. Dendritic branches are not just passive conduits to the soma. They're active computational subunits. Each branch integrates its own set of inputs, and because the integration is nonlinear, it can generate its own local spikes. That means each branch can potentially act like a little processing unit, an elementary memory module. And if that's true, it suggests a second layer of computation, as it were, inside a neuron. The synapse is then no longer the smallest meaningful piece, instead the branches. Now let's contrast that with the classical picture. For decades, memory was explained through Hebbian synaptic plasticity, and it still is. Neurons that fire together wire together. And Rall's 1962 cable theory gave us the math for how signals spread in dendrites, but the focus was largely on passive properties. Later studies, like the paper here by Segev and Rall in 1998, recognized dendrites could be excitable and produce spikes, but the neuron was still treated as a single unit. And what's new here is the claim that branches can function independently enough to be the true sites of memory allocation. And it's not all speculation. The authors pull from a lot of converging evidence. Modeling work from Mel in the 90s showed that clustering of active synapses, especially with NMDA receptor activation, boosts nonlinear problem solving. And Poirazi and Mel in 2001 modeled how the arrangement of inputs in time and space impacts capacity. On the experimental side, Frank et al. And Harris show synapse clustering both functionally and anatomically. And Polsky et al., Spruceton, Lavzin show dendritic spikes and local depolarizations can drive plasticity. Branco and Hauser frame dendritic branches as functional units. So all of this supports the idea that memory traces might live at the dendritic level. So an important point is that you cannot isolate synaptic changes from dendritic ones. They're coupled. For example, dendritic spikes enhance LTP. Plasticity can be specific to a compartment or involve cooperative interactions across nearby synapses, and excitable changes can be localized to part of the dendritic tree. In other words, the container, the dendrite, shapes how the contents, the synapses, behave. So computationally, the compartmentalization leads to a greater capacity for potential engrams with which an individual neuron can be involved. We'll get a bit more about that in a moment. This is the first figure from the paper. So figure 1 gives us a visual here where you can see clusters of potentiated synapses sitting on a single branch. And when those clustered inputs are activated together, they can produce nonlinear events like NMDA spikes. And that branch then behaves differently. It's primed to participate in the recall of a particular memory. So cluster-specific changes directly affect current ratios in the engram cells versus non-engram cells. Dendritic spikes aren't just small ripples. They can trigger bursts at the soma and elevate calcium locally and drive synaptic changes. Usually, dendritic and somatic activity are tightly correlated, but Franconi et al. Found behavior-specific calcium transients confined to certain branches. That means two branches of the same neuron could be representing totally different behavioral information at the same time. Now we know that spines grow and disappear with learning. Spatially, they tend to form or disappear near other spines, and partly that's because presynaptic and postsynaptic elements are more likely to connect again in the same vicinity. Actually, I had to wonder, how much does this represent a novel aspect of neural computation versus to what degree are the effects already incorporated in our representations of neural computation and plasticity? So perhaps these hotspots are expected as a result of axon dendrite locations in each other's proximity, and some of the computational benefits that are talked about here could already be accounted for by mechanisms like the overlapping NMDA PSPs. I say this mainly because in modeling work that I was doing yesterday and day before, these are very typical things that I see anyway, and then I wonder, oh, I can get the same effect just using those overlapping NMDA PSPs. Still, the fact that clustering emerges naturally may actually help the dendritic engram hypothesis. It provides a biologically plausible path. So, I guess here's where it gets fun. If two memories land on the same branch close in time, potentiating one can also potentiate the other, and this is called co-allocation. Kai et al. And Segal et al. Show that temporally linked memories often share clusters. In modeling, you need both dendritic excitability and plasticity of this kind for this kind of linking to work. All right, and here we're at the next figure in the paper. And I should point out that if you've been reading the paper, try not to get confused because they actually flipped A and B. I see here they're discussing the HLTD, and that's at A. It's this one here. Whereas they just they gave it the wrong label since they say B here. So I've just given it the labels they're supposed to have. This figure outlines the molecular processes that are hypothesized to support heterosynaptic plasticity. So from one synapse to another on the same dendritic branch. When one spine is strongly activated, calcium influx triggers cascades, CAMK2 activation, BDNF, that's brain-derived neurotrophic factor release, and that in particular, I mean that, and then of that, particularly the small I don't know how to say those, GTPases or GTPases, a group of proteins. Anyway, it's a collection of them. They're listed right here. The ones that are relevant here, RAS, Rho A1, RAC1, and ARC. These proteins can spread to neighboring synapses. So I've circled that here. You see the spread going on, and here's the spread again. The reason why this one's green and this one's red is because this one, oh, maybe, did I flip it around because I was following their A and B? No, I didn't. Nope, this is the LTP case, and that's the LTD case. So in this case, yeah, if the neighboring one in this case here is active, then you might get, inactive, that's this one here. Then you might get heterosynaptic LTD, so a spine shrinkage or removal of AMPA receptors. And if the neighbor is active, then you might get heteroassociative LTP, actin remodeling spine growth. This means that the plasticity state of a branch is shaped by the pattern of active and inactive neighbors, and not just the cell itself or the synapse itself. By the way, I wanted to say that if you have questions, please just blurt them out or comments, because I can't currently see if someone's holding up their hand. Now, from a computational view, dendritic branches are subunits, and the requirements for that include the ability to generate dendritic spikes and branch-specific inhibition to keep branches functionally separate. If you have that, then the computational effects include modulating synchronization by regulating dendritic spike spread, dendritic spike induction of plasticity, and treating it as a functional unit of spike integration and plasticity. Nonlinear interactions between neighboring synapses that act cooperatively can be supported by several mechanisms, including the lateral spread of messengers that we just discussed, or a presynaptic nonspecific diffusion of a messenger. And in that case, the effects include more complex computations are possible per neuron, a local lowering of plasticity thresholds via molecular spread, and cooperative plasticity among neighboring synapses, usually within an area of about 10 micrometers on the same dendrite. Now, local positive feedback loops can stabilize memory traces. NMDA receptors that are acting as coincidence detectors, CAMK2 binding that keeps NMDA channels open longer, and BDNF secretion that boosts plasticity are involved there. And also crosstalk, or heterosynaptic crosstalk, comes from those small GTPases that diffuse to the neighboring spines, triggering growth or stabilization. And those processes can happen within milliseconds to minutes. So branches are very dynamic environments. On the right here, this is a diagram that is showing this idea, this hypothesis of tag and capture. So the tag and capture, this hypothesis said that active synapses set a tag and later grab plasticity-related proteins that are coming back from the soma. And if you apply that to heteroassociative LTD, you can tag inactive synapses for weakening, either via local processes or signals from the nucleus. And my question then would be if this LTD is more spatially restricted than the classic STDP would suggest. And that's not yet clear to me, because the the modeling of STDP as we normally see this, coming from B and Poo and other publications, typically already includes a type of LTP that is that gives you kind of the same effect, and whether it's spatially restricted or not, that is still something that I don't quite understand. If you treat branches as subunits, then models show up to a 40 times boost in capacity. And that's because arrangement matters more than strength. So clustering encodes more distinct patterns. There's also an optimization angle to this. Binary-like weights, which we saw support of in the Dorkenwald paper that was recently presented. They can help avoid catastrophic forgetting, and dendritic segregation allows self-organization within a neuron. On the biological side, spatially patterned protein synthesis lets you selectively encode or forget events, even if they involve the same neural ensemble. Now we're at the third figure from the paper. This one is presenting us with a number of different computational models that can be part of the toolbox, examples of how dendritic computation, or these dendritic segregations into compartmental or units, can be applied. And in the first case here in A, this is an example of two-layer neuron models where branches are nonlinear nodes. And notice that this can still use your typical run-of-the-mill integrate and fire method of modeling neurons, except that you're now splitting up the cell into these two separate layers. So you've got yourself a little network in a sense. Then another approach or another application has been that in hippocampal pathway models, using dendritic spikes for online learning, they demonstrated that using a model where you have these dendrites, each of which spike independently. So this is real-time adaptation to new experiences. And Wu and Mel worked out some learning rules that were optimized for this, using independently thresholded dendritic branches and using a combination of LTP, that's for the learning, and homeostatic depression for forgetting. And again, effectively binary valued synapses. This keeps coming up, and it's kind of exciting given the other paper we discussed. Models that combine protein synthesis with excitability changes are used to explain why memories within about 5 hours tend to co-allocate on the same branch. So these are just a few examples of directly applying this separation into dendritic branch calculations or computations, actually applying that in models. And again, it's not particularly difficult to do. It's basically still like using integrate and fire neurons. Now, it's not just pyramidal cells. Inhibitory interneurons, like the fast-spiking basket cells, also show dendritic nonlinearities. And this means they, too, can act as two-stage integrators, potentially shaping how engrams are formed and stabilized in a network. And Silivaki et al. Demonstrated both supralinear and sublinear integration in hippocampal and PFC basket cell reconstructions using this approach. Now, we're almost at the end already, so this has actually been a very short journal club presentation. Stepping back, the dendritic engram model blends Hebbian plasticity, branch-specific cooperation or cooperative effects, and then the somatic level integrates all the feed-forward and feedback signals, but the fine-grained storage happens locally. So that's kind of a top-level way to think about this. Now, the authors also suggest some future investigations. So it suggests experimental goals that are not just mapping which neurons are active, but which branches are storing a trace. And this is shown in figure 4. High-resolution time-lapse imaging of dendritic segments before and after learning is one good approach, and this is a bit similar to what we saw in El-Bustani. Branch-targeted molecular tools can be used to manipulate plasticity locally, and tracking of plasticity-related proteins across time. And then, of course, computational modeling to test the limits of capacity and stability. And then I want to add some thoughts because, I don't have any particular doubts about clustering of synapses or of dendritic nonlinear effects. So that's not really an issue, but I'm wondering how much this actually changes how we represent circuits and how we model them. And some things, well, there are some things I wonder about in terms of stability and robustness. So can branch level changes be stable and robust over time? And another thing is that point neuron models do really well. They still do very well in many tasks. So the question is, do we really need dendritic subunits to explain behavior? And then I think there's still not really a slam dunk case. It's not clear to me is the output of clustered synapses meaningfully different from distributed ones in real circuits. So in a functional sense, the things you can model with it. These are all testable questions, I think, and answering them could tell us whether dendritic branches really deserve to be called the fundamental units of memory. Now, I've already reached the end of this relatively short presentation, but I would love to hear your thoughts on introducing the dendritic engram as a concept or not, and what your thoughts are. Go ahead, Ken.
That was great, Randall. I think this is a really important paper, review paper. Just a couple of thoughts off the top of my head. One is that the number of outputs for a single neuron would be on the order of 10,000, right?
Sure.
The number of outputs for a single branch is 1.
1, yeah.
Okay, so I think that somehow puts something into perspective here in terms of I don't know what constitutes a branch. A branch might be a typical pyramidal cell might have about 10, 12 branches, apical and basal branches. I'm not exactly sure where the cutoff is, but I think that does put some perspective on. There are a bunch of studies that kind of argue that this is not that important of a fact. These are not compelling studies. One of the ones I'm thinking of is there was one that we covered in a journal club a long time ago where they used a laser to cut different dendritic branches off. And they were asking the question, how does the visual receptive field change when you cut off dendritic branches? And my recollection is that for the most part, it didn't change that much. Which again, it's not a big thing, but you could imagine that would be somewhat of a null effect here. Is there a experimental? The modeling work is very compelling. It's like the brain could use this, no question, right? And it could be storing more information or doing, the equivalent of having twice as many layers or something. But what is from your reading? What is the most compelling evidence experimentally that this clustering is having a major influence on the computation that's being done?
I'm going to answer the question slightly differently because I want to give you a little bit of my impression of this paper. I got the impression that the experimental studies mentioned did a fairly good job of indicating that clustering occurs and that heteroassociative plasticity is possible. What I didn't really get is exactly what you're saying. I didn't get the sense that there is strong experimental evidence of something that only works due to treating the dendrites, the dendritic branches, as units of a sort. And the same thing happens, and this is why I had that critique at the end, when I look at it from the, say, computational modeling point of view. Like, I want to achieve a thing that can, learn relationships between different engrams, and then I see things that are being explained here, for example, as heteroassociative plasticity, happening even without directly modeling these molecular cascades and the effect on neighboring synapses, because this clustering that, and I don't know, okay, I'm saying because, but this is my hypothesis here, because I think that the clustering that they're observing, which then facilitates this heteroassociative plasticity, is a natural outcome, at least in the way I'm modeling, of engrams that tend to, on occasion, or where inputs on occasion co-occur. And then the effect of this heteroassociative plasticity actually becomes part of the learning, part of the presentation of stimuli, no matter whether you're depending entirely on this clustering or not, because the clustering, in a sense, part of the clustering happens by itself automatically due to the proximity and spiking of inputs. And to another degree, just the co-occurrence of spikes leads to this association between the different engrams. I think there is a potential here for an effect that, for instance, could tie into the presynaptic learning that we've also looked at in other papers, which, you could see how that can be facilitated by heterosynaptic plasticity. I keep saying heterosociative. But it's just not clear to me that this effect is strong, or that this is something that I've observed where, this would be different in models. And that's just kind of a gut feeling, really. Go ahead.
Can I ask, how should we think of this model? Is it in a way about reconstructing the exact same memory each time that dendrite is triggered, and it's replaying kind of the same sequence of action potentials from the same neurons? Or is it that it's almost like a pointer, and it's like rebuilding a memory from scratch each time? So every time I re-experience the memory, it could be slightly different. How do you tend to kind of think of the way this describes memory?
I don't think it really points in one direction or another, because changes or plasticity, I think, are always possible. So every time you have stimulus, you can have change. But at the same time, what is there is being used as a discriminator or feature detector, a part of a circuit that's established. I don't know, Ken, do you have a better answer for this? Or Ariel? Or anyone?
I don't.
But I don't see a huge distinction here between this and your regular neuron model, in the sense that those models also incorporate both aspects all the time. Maybe I'm too critical about this. I don't know.
Well, I will offer one. I'm not sure if this is related, but from a connectomic point of view, these large-scale connectivity graphs, they have both the neuron-to-neuron synaptic connectivity and the branch-level connectivity. And in fact, this Dorkenwald paper compared those two, if I recall. I'm trying to recall exactly what they got, but my recollection, please correct me if I'm wrong about this, was that synapses with the same pre and postsynaptic cell were correlated. And if they bunched those into ones that were close in presumably close on a dendritic tree versus far, meaning on separate dendritic trees, they were still correlated, and I think there wasn't a statistical difference between them. Is that your recollection of that paper?
Yeah, they were correlated both in volume and in size, I mean, and they were also correlated in terms of what is the level of AMPA, was it, or AMPA NMDA ratio? Was that it?
I'm talking about the Dorkenwald one, which is pure structural connectomics.
Oh, okay, sorry. Yeah.
But
yeah, I don't quite recall.
Yeah, I'm not sure if they. It'd be a difficult experiment without that. To do a true functional experiment, I think, with matched synapses that are far apart. In any case, this is the kind of thing that might come out of connectomic studies, is what I'm saying. Because if there is a signature of really like this is a dendritic branch type of thing, and two dendritic branches might as well be on two different neurons. If that's the strong case that is being made, which I don't think it is, but that's kind of that one of the strongest cases would be two branches on the same neuron might as well not be on the same neuron, right?
But in a study like Dorkenwald, I think this was still difficult to decide because we couldn't even be sure that all of the dendrites to a particular axon were within the volume that had been reconstructed. Or were we?
I don't think that would matter, though, because if you had two synapses that had the same pre and postsynaptic cell, you could ask whether those two synapses are on the same dendritic branch or on two different dendritic branches on the postsynaptic cell.
Sorry, what I meant to say is that even if you see that a pre and postsynaptic neuron make a lot of synapses that happen to be on the same dendritic branch, you might not know if they're also on other branches to the same postsynaptic neuron if those dendritic branches fall outside the volume.
Absolutely. I'm just talking about a statistics of the synapses that you really do know the pre and postsynaptic partners.
Right.
So
sure.
yeah. And of course, this may be very different from different neuron types. It might be one. Maybe neurons in layer 2, 3 of the visual cortex could care less about dendritic branches, but in barrel cortex in layer 5, this is the most important thing that as part of the computation. It's yeah.
But you mean you have to have both, right? You need to have a structural investigation, which is, this paper was mostly about, here's all the evidence that there is a clustering of synapses, which is kind of like one step towards this structural information about, say, do pre and postsynaptic neurons that typically connect on the same dendritic branch. But in addition, you also need modeling that can support that there is a functional distinction of relevance between what these pre-synaptic neurons can accomplish if they're on the same dendritic branch versus not.
Yeah, and I think that is the key point there, okay? Because as you were saying, if I'm interpreting it correctly, you would expect this, right? You would expect clustering. You would expect clustering to happen, even if the brain didn't want this to happen. It's kind of like leakage would accidentally cause clustering to happen.
Yeah, you would absolutely get clustering, and that's whether or not you depend entirely on this, transfer of proteins through that part of the dendritic branch, or if it happens over some time because of pre and post synaptic spiking. You'd get some clustering. You would get maybe more clustering in one case than in the other, but you would tend to expect to see clustering happen.
Yeah. So what I'm saying is there's a more core assumption that dendritic branches are the fundamental unit of learning.
Yeah.
And if a dendritic branch is the fundamental unit of learning, then in the strongest sense, if it is, then synapses on two different dendritic branches on the same neuron shouldn't have a correlation more than on two different neurons. I think that's too strong. We already have evidence against it. But essentially that it rules out the strongest case that some of these models would be making.
Mm. The funny thing is that they actually show that right here in the model. They've already got their clustering on multiple dendritic branches to the postsynaptic cell for memories 1 and memory 2. Yeah. So even in the model, they're not necessarily assuming that those are distinct. Yeah. But yeah, if you could keep them, if you could show that somewhere they are really separated that way, then you could imagine that the nonlinearity from one branch to the main trunk and onto the soma, that has a significant impact. Yeah.
There was one sentence in the paper that I found very intriguing, but I didn't look into the references. It is a review paper. I was wondering if you looked into it. They said that there was some modeling work that said you could store under some circumstances 40 times as many memories.
I should have looked that one up. Yeah, I saw that as well. I even put that number in the slides here, but I didn't look up the model in question to find out how they came up with that.
Okay. Is there, along those lines, is there any kind of just really stunning difference from a modeling perspective that would make this way more powerful and would essentially throw a wrench in any modeling that really didn't assume this?
It depends on what your normal type of modeling is, because I think so. The reason I didn't look up that reference was because I figured, okay, yes, if you treat each branch as a separate unit, and connections really only go to one branch or another and not across branches, then they will form to a degree separate units, and that does give you some additional discriminatory power. So I didn't know why 40, but it kind of makes sense. But this will only make a difference to your modeling if your normal way of modeling is to treat it as a point neuron, because then you really have, what you're seeing here, you've got really a little network instead of a single point neuron then, right? But that's, at least for me, that's not how I normally model. So since I'm already not treating them as point neurons, what I'm doing is it already kind of subsumes the effect, I think. So the neurons that we're usually using right now are using integrate and fire, but they are compartmentalized. So it's already taking that into account anyway. Yeah, go ahead.
Yeah, I don't want to dominate with questions here, but so one of the other things that I am very curious about when I read any paper like this is when they're talking about different learning mechanisms that are not synaptic, that might not be visible in electron microscopy. And I think they talked about, they definitely talked about intrinsic excitability of neurons and intrinsic excitability of dendritic branches. Makes sense. But they also hinted at the possibility of changing dendritic branch excitability. Did they give any examples of where that's been seen, that it's like this dendritic branch for a long time period, not the short time period that they're talking about that might be doing this clustering, but for a long-term memory that changes the excitability of a dendritic branch? Because that would be really earth-shattering from my perspective.
You mean, did they show experimental support for that?
Yeah.
I do not recall whether they did. Maybe someone else here who's read the paper.
Because you see what I'm saying. It's like,
Yeah.
it would also be very important if they said this neuron became more excitable. Its bias essentially changed. But the literature that we've been looking at kind of, at least my reading, typically says that this increase of excitability is transient, and it's used to pick out neurons that might form an engram in a particular time window. And it's not something where you stamp a new excitability to store a memory from a long-term perspective. But if you could do that on a dendritic branch level, that would be a real important thing to understand. For example, adding new ion channels to a branch, and making that whole branch more excitable, as opposed to just putting stronger synapses on a branch, but the branch itself has the same number of
How is
channels.
this functionally different from something like early LTP?
Well, it would be long-term.
Okay, how is it functionally different from late LTP?
From my perspective, the big difference is that you can see it in the electron microscope.
Okay, so a particular anatomical or molecular mechanistic support for what is already functionally being modeled as part of long-term LTP or late LTP, right?
Yeah.
Okay, so it doesn't really change anything in our functional understanding of how the neuron model would work. It just would give some support to it.
It would be a hidden variable that connectomics,
Could pick
EM-based
up on.
connectomics
Okay,
would be
yeah.
blind to. Yeah.
Yeah, okay. Yeah, this is all very relevant if anyone wants to make sure to decode memory that includes that temporal window. Yeah. Maybe that's something to focus on, is different techniques that pick up on different temporal windows of memory. And so then the decoding can include early phase or medium term changes, as opposed to what electron microscopy can pick up on. Yeah.
Yeah, but I'm also talking about long term. So it's like if you use something like Lycon to look at the different ion channel distributions on dendritic branches, and you saw, oh, this whole branch is filled with a particular type of calcium
So
channel that is going to cause more excitability.
but you're assuming that this, whatever it is that you can pick up on that would show that it's more excitable, that it would be uncorrelated or not correlated with changes in synapses or structure that you could see under an electron microscope?
I'm not assuming that, but the implication would be that that could be a possibility.
Okay, if there is such a thing that is not correlated with the changes you could see under EM, then that would indeed be something new to worry about. Yep.
Yeah.
I don't know of such a thing. But again, others here may have read claims. Are there any further comments or questions about this paper or about the topic that we're talking about right now? If not, then the only thing that remains is for me to thank everyone who showed up for this journal club, for being here, joining us. Yeah, thanks, Jacob. Yeah, I'll just, in ending, I will add that when I started reading this review paper, I was really hoping that we would end up with something dramatically new and interesting that I would then have to go and implement in my models, because it would make a huge difference. And I cannot say that I came away with that impression. But maybe I just need to read more papers about dendritic computation and clustering and branch units, etc. Thanks, everyone. Have a great day.
Yeah. Thanks, Randall.
Thank you.
Bye-bye.